I'm your tree pest, and I will continue to be your tree pest.
No, you're not.
Oh, okay.
Yeah.
You just call me that when I email you stuff. I'll remember that. So today we have Dr. Enrico Bonello, who is in the Department of Plant Pathology, and he's going to expand our horizons with mycorrhizae information. So
Wow.
with that, I'm going to stop sharing, and it's all yours.
Well, that's a challenge. Good morning, everyone. I am going to start with a little video that I think will just whet your appetite a little bit. I don't know if you've seen this before, but it's a short National Geographic produced video. And you guys can all see this yellow screen. Kathy?
Yes,
Yeah.
You're good to go.
Okay. So I'm going to run the video and then we're going to delve into what mycorrhizae are in more detail, okay?
Ouch! What do you think you're doing?
The idea of talking trees has been capturing the human imagination for generations.
Did you say something?
My bark is worse than my bite.
Okay, so maybe they don't talk to us, but it turns out trees can talk to each other.
The trees are speaking to each other.
But that does beg the question, what do trees have to talk about? And can we learn to speak their language? Underneath the soil, a vast and interconnected network of life links the trees through their root systems. But they can't talk to each other without help. The whole process starts with hub trees.
Sorry, folks, I'm going to eliminate those closed captions, if you don't mind, because they're covering the important parts of the video. So let me go again here. I'm going to go back a little bit.
The whole process starts with hub trees, the oldest and tallest trees in the forest. Hub trees have greater access to sunlight, and through the process of photosynthesis...
I think I missed, sorry, let me do it again. I think this is where we were, actually. There we go.
Underneath the soil, a vast and interconnected network of life links the trees through their root systems. But they can't talk to each other without help. The whole process starts with hub trees, the oldest and tallest trees in the forest. Hub trees have greater access to sunlight, and through the process of photosynthesis, end up producing more sugar than they actually need. Underground, fungi need sugar to survive. Most of their bodies are made up of a mass of threads called mycelium. They grow within the root system of trees to absorb the excess sugar. In return, the mycelium provides the tree with the nutrients it needs from the soil. This symbiotic relationship is known as mycorrhiza, which stems from the Greek words for fungus and root. These tree-fungi relationships connect the trees in the forest together, forming an underground communication network to exchange water and nutrients, to nurture their seedlings, and even send warning signals when under threat. So, how many trees are really talking to each other? To get a better picture of these forest relationships, a team of researchers used DNA analysis to map a fungal network in a patch of Canadian forest. Remarkably, they found that one tree was connected to 47 other trees. Their models also showed that when hub trees were removed, it would cause more connections to be lost than if trees were simply removed randomly. Studying these kinds of underground exchanges will play a vital role in creating stronger, more resilient forests for the future. So even though we might not be able to talk to trees, at least we can still keep trying to understand their language. Who knows what they might say?
Right, so that's the end of the video. I'm going to stop share that and get on to the PowerPoint presentation, which is down here. Actually, oops. Bear with me for a second. What's going on here? Share screen. Stop share. Okay. Oh, there we go. Let me move this out of the way. So sorry about these technical issues. So as you've seen in the video, mycorrhizae are a symbiosis between fungi and plant roots. And also, as you've seen in the video, it comes from the Greek. I like to start with the meaning of words because they actually signify something, as a matter of fact. So it comes from the Greek, myces for fungus and rhiza for root. And that in itself already gives you an idea that it's a fusion of these two things that create mycorrhizas. It's also symbiosis. Now, symbiosis in the common language is often taken as meaning mutualism. In other words, an association that's beneficial to both partners. But in reality, the term symbiosis simply means living together. And so, for example, even pathogenic interactions where you have a plant and a fungal pathogen is a symbiosis. It's just not mutualistic, okay? So that's a critical difference. So really a complete definition of mycorrhizal symbiosis is that it's a mutually beneficial, so it benefits both partners, anatomically intimate. So these things have to be in close contact. In fact, they grow into each other. Biotrophic associations, and biotrophic means that the fungus needs a live host plant to generate a mycorrhizal association. You cannot have a mycorrhizal association where the plant is dead, right? When you have fungi attacking dead plant material, that's a saprotrophic association. So it's a biotrophic association between fine, also called feeder roots of higher plants, and some special soil-borne fungi. Okay, so that encapsulates what the mycorrhizal symbiosis is. You need to have all those elements. Has to be mutually beneficial, has to be intimate and biotrophic to be a mycorrhizal symbiosis. Now, I want to start out the conversation today by showing you that there's a very close overlap between the origin of land plants. So here you have the different epochs, you know, the geological eras or epochs. And land plants sort of appeared first during the Silurian epoch. At the same time, you have the first records of what are called VAM fungi. This stands for vesicular or vascular mycorrhizal fungi. We'll see that what that means a little later. And these days, by the way, the V has been dropped and they're called AM fungi. Anyway, there's a very close overlap between the appearance of land plants and also the origin of these fungi, which suggests that the appearance of land plants was actually mediated by the appearance of these mycorrhizal fungi. In other words, colonization of land by plants from water environments was facilitated, if not made possible, by these fungi. Here's some fossil records showing these associations. So this is 400 million year old fossil record of mycorrhizal-like associations in this particular plant, Egleophyton major, a rhizome of this plant. What you see here are these agglomerates of hyphae, these thread-like structures that fungi produce. inside their cells, and also vesicle-like structures, hence the name vesicular or vascular mycorrhizal fungi within the same cells. So again, the fossil record shows that these associations were present early on in the evolution of plants and the ability of plants to colonize land environments. So they're clearly very important, not only from what we know now from function of these associations in nature, but also from the fossil record. So, and by the way, if you're interested in all possible details regarding mycorrhizae, you can go to this site. It's pretty easy to remember, mycorrhizals.info, and you can find all sorts of information on mycorrhizal associations. And I'll be going into some detail about the categories that are known these days. And so, but anyway, that's a reference for you guys if you're interested in a lot more information than I can present today. So what I'll say today is I have this is a summary table that's kind of busy. But what I want to say is that I'll be talking today about two types. Let's see if I can get my, maybe I can change my pointer. Oh, here we go. This one. It's a pointer. Okay. I'll be talking about these two mycorrhizal association types in some detail today, but there are all these other types. So let me just talk about, for example, the first VAM. Here are some traits that are essentially important or essential for an association to be called a VAM. So the hyphae that I mentioned even earlier, hyphae are these thread-like structures that fungi produce in the soil, for example, as you've seen in the video. The vesicular vascular mycorrhizal fungi have hyphae that are not septate, so they don't have subdivisions within them. They're like essentially these very long, very thin tubes without cross walls in them. The hyphae of these fungi penetrate inside the cells of the host, and they produce these very particular structures called arbuscules, which gives the name to the group, right? So it's vesicular arbuscular mycorrhizal fungi. Sometimes they produce vesicles, sometimes they don't. And essentially they affect all plant groups. So I won't go into the groups of fungi that are involved here because that's too specialized. But suffices to say that these particular associations involve all different major plant groups that we know that have been classified. Ectomycorrhizal fungi, on the other hand, so ectomycorrhizal, ECM, are characterized by fungi that produce hyphae that are septated. So they have these cross walls in those tubes. They don't penetrate the host. They don't produce arbuscules, but they produce two other structures that are very important for their classification. One is called the mantle or sheath, and the other one is called the hartignet. And I'll show you very briefly what these structures look like. Now, the ectomycorrhizal associations really affect only gymnosperms and angiosperms, right? So gymnosperms, think your conifer trees. Angiosperms is all the flowering plants, dicots, monocots, and so on. Okay, so they're also pretty widespread. There are other groups that only affect specific groups of plants. For example, the Ericales for the arbutoid type, or the Orchidaceae for the orchid type. But I don't have time to talk about these specific types today. I just don't have time. So I'll focus on the first two, and I'll start with the Ectomycorrhizaes. And I'll show you first a few micrographs that I took during my PhD work. We're talking about prehistory here, like the dinosaurs. But what I was doing there is I was growing pine seedlings in petri dishes, and I was inoculating them with an ectomycorrhizal fungus, and I was following their development on the root system. So here you have a bifurcating feeder root of the seedling, and you see these threads that surround that root tip. those are the hyphae of the fungus. And as you can see, they really attempt to congregate on the root tip. They essentially sense the presence of the root, and they grow toward the root, and they start infecting the root, just like a pathogen would in many ways. But the difference, though, is that these fungi start producing the mantle that I was talking about earlier, a sheath. So this is a structure that is made up of fungal material that essentially encases the root tip in fungal material. Here's a 3D perspective using electron microscopy that shows you, these are roots of eucalyptus. These are primary roots of eucalyptus. So you see the root hairs here. You see initially hyphae coming in contact with the root, and then over time, the fungal material grows on the surface of the root, and it completely encases it in this thing called a mantle or sheath. That's why it's called Ectomycorrhizae, because it's outside the root. And it's also outside the cells, as you will see in a moment. Now, these associations, which are typical of trees for the most part, can take on very interesting morphologies and features that sometimes can help identify the particular fungal species associated with the particular tree species, okay? So some of them are dark in color, some of them are sort of yellow-orangish, some of them are very smooth and shiny, others are more matte, or some of them are covered in these hyphae. And these are the hyphae that you saw in that video schematically. These are called extramatrical hyphae. So these are hyphae that grow, well, they grow from the fungus into the root system, but then also from the root system out into the soil, okay? And we will see the function of these structures is essentially to extend the volume of soil that the root system can explore, okay? Like extensions of the root system, basically. So that, as we will see later, the fungal mycelium absorbs water and nutrients from the soil and conveys it to the roots, right? So that's a benefit to the plant. And vice versa, the plant gives sugars essentially and amino acids to the fungus so that it can grow and complete its life cycle, okay? Now, I like this slide because it shows that at the very early stage, this is a seedling of a pine tree. The fungus itself, which in this case is this mushroom, can have can be a major draw on the photosynthetic ability of the plant to provide carbon or sugars to the fungus. So all the white stuff that you see here, this is all fungal material that's growing on the root tips. And then it acquires enough energy to be able to produce this fruiting body. This is a fungus called Lactaria laccata, and it's actually an edible fungus, an edible mushroom. But just look at the difference in biomass between the fungus and the root system. It already gives you an idea that these kinds of fungi associated with the roots of plants actually are parasitic on the plant. They're actually absorbing nutrients from the plant. In fact, there are estimates that in a forest environment, about between 10 and 15% of the carbon that's fixed by the plants, so the sugars that come from photosynthesis, end up in fungal material in the soil, okay? So if you think about it, it is a major expense for the tree or for the plant in general, but the plant only does it because it gets something in return, and that's the nutrients, the mineral nutrients, and the water that I've already described, and that was also showed in the video quite nicely. So anatomically, Ectomycorrhizaes, so this is a root tip, okay? Anatomically, the Ectomycorrhizaes are characterized by a mantle, as I showed you in that initial table, and then a heartic net. and harticnet will be more apparent in the next slide, but essentially you have the fungus that grows in between the cortical cells of the root. So the fungus never penetrates the host cells. It always stays outside the host cells, which is a very interesting phenomenon, but that's where the nutrient exchanges occur between the plant and the fungus. Okay, here's another perspective. So you have the mantle on the outside. This is all fungal material. As you can see, the hyphae change morphology completely. They look more like cells that you would find in a plant. And then you have this fungal material that grows in between the cortical cells. Essentially, the fungal material envelops the cortical cells completely. And that's, again, to maximize the surface area where the nutrients can be exchanged between, and this other kinds of signals between the plant and the fungus. Okay. Here's another perspective. If you take even a hand section, which means if you take a root and cut it with a razor blade, and if you're very careful and cut it pretty thin, you can put it under the microscope and you can see the mantle and then the harticnet. So it's called a net because it looks like a net, and it's called harticnet in honor of the father of forest pathology, really, a German forest pathologist from the 1800s who first discovered this phenomenon. So again, Ectomycorrhizals have a mantle and a harticnet. I'll skip this other slide here, image on the right. So normally I would have questions at this point, but it's a little difficult through a webinar. So if you have questions, hold them, jot them down, and then you can ask them at the end. So I'll move on and talk about Endomycorrhizaes, which are the VAMs that I mentioned earlier. So the vesicular arbuscular mycorrhizaes. And as I said, these days are actually called arbuscular mycorrhizaes. I leave the name vesicular because some people still refer to them as VAMs. So here's the situation is very different. So here you have a cross section of a root, okay, a longitudinal cross section. So the length of the root is this way. And you have an epidermis. So the external surface layer of cells is the epidermis. The fungus comes in contact with the root like the other ones do, but then at some point it penetrates into the host cells. Okay, that's why they're called endomycorrhizaes. They proliferate inside the cell just like a pathogen would, but they don't make the plant sick. In fact, they are very beneficial to the plant. Sometimes they produce vesicles, as you can see here. But the key feature is the production of these arbuscules. These are structures that technically are called haustoria that grow inside the host cells. And they're called arbuscules because the word comes from the Latin for tiny tree. And that's what they look like. So if you can imagine in this particular micrograph, a cell wall of a plant running through here. You cannot see it because of the microscopy that's been done here. On the outside of the cell, you have a hypha growing alongside, just like it shows right here. Okay. And then the fungus decides that it wants to penetrate inside the whole cell. So here's a constriction point because that's where the hypha passes through the cell wall. And then once inside the cell wall, it proliferates in this very ornate manner. And the reason or the function of this high proliferation of hyphal structures is that it's maximizing its surface area of contact with the host cell, again, to maximize the exchange of nutrients and other signals. Okay, I hope this is fairly clear. Here's another perspective using scanning electron microscopy, where you get even a better idea of why these things are called tiny trees, like the arbuscules. But again, you get this idea that it's a very, extensive branching of these structures inside the cells because it's trying to maximize the surface area of contact. So that was all about the morphology and anatomy of these different associations. I want to talk a little bit about taxonomy and specificity. I see somebody is raising a hand, but I'll wait until the end if you don't mind. just note down your question for later. So this slide sort of tries to show on a global level what the prevalence of the various different of the various kinds of mycorrhizal associations is. And as you can see, the majority of plants, 50%, are affected by the arbuscular mycorrhizas that I've just talked about. The ectomycorrhizas really affect about 20%, are sort of included in this 20% that also includes orchid, ericoid types, and so on. So they're definitely a minority. The reason that VAM are a majority is because they affect essentially all herbaceous plants, all crop plants, all grasses, all forbs, most shrubs, and also some trees sometimes. So they're very widespread. If you look at ornamental plants, and I apologize about this, the presence of this thing here. There we go. This is a partial list of ornamental plants. And when I say plants, I actually mean trees for the most part, because that's what I work with. You will see that, most plants or most trees are either one or the other type, but not completely very often. And some of them can be both just by their nature. So alders, eucalyptus, willows, and so on, cottonwood, cedar, and cryptomeria can be both ecto and endomycorrhizal at the same time, or depending on the different conditions where they grow. Others are predominantly endomycorrhizal or ectomycorrhizal. So some of the most iconic tree genera like pines, those are all ectomycorrhizal. Oaks are all ectomycorrhizal. Beech, same thing, and so on. As far as diversity of ectomycorrhizal fungi, so ectomycorrhizal fungi are actually more specific to their hosts, and that means that being more specific to their hosts, there's a wider diversity of ectomycorrhizal fungi associating with those different hosts. So as a matter of fact, globally, it is estimated that the total number of ectomycorrhizal fungal species is between 5,000 and 6,000, and perhaps even more. This is kind of an old data set, but it's probably not far from reality, but it could be a little less or a little more. Even at the very local level, you can have quite a high level of diversity. For example, you can take a single forest type like a pine plantation and like a pine monoculture, and you can have 50 to 30 different species of ectomycorrhizal fungi in a single hectare. I remind you that hectare is about 2 acres, okay? And even at the micro level, if you look at the roots adjacent to each other, you can have more than 2 or 3 different types. So here is an example of a bishop pine root. Bishop pines grow on the coast in California. And you can see very clearly that even within this very tiny space, there are 3 different types of mycorrhizal associations based on simply on the morphology of these structures. Here's a typical ectomycorrhizal fungus. This is a boletoid, so it's the same family as porcini mushrooms. If you are a fungal aficionado as far as cuisine is concerned, this is not very good, but it's an important ectomycorrhizal fungus in those bishop pine forests that I mentioned just a second ago. And that's very different, as I will come back to it, but it's very different for endomycorrhizal fungi because the level of specificity there is very low. In fact, it's estimated that there's only about 200 species across the globe of different endomycorrhizal fungi. So I want to come back to these ecological interactions because I think it's one of the more interesting aspects. So there's 2 types of fungal partners in symbiosis with plants. Remember, I mentioned this at the beginning, and it can range all the way from pathogen examples. Think about potato late blight or chestnut blight, which are pure parasites, right? All the way to the mutualists. So with the mutualists, you have associations in which both of them gain nutrients from each other, or they gain nutrients from the host, but they give something back, some other kind of benefit. With plants and fungi, these are the two most important examples. So the mycorrhizal fungi, which are the ones we've been talking about, and then the lichen fungi. So the lichens, you may know or may not know, are symbiotic mutualistic associations between some special fungi and unicellular algae, or also they are also called cyanobacteria. Now, morphologically, they're very different, mycorrhizal associations and lichens, but functionally, they're the same thing. The plant, either a higher plant or the algal associate, provides sugar to the fungi through photosynthesis, and the fungi supply nutrients and water to the plants. So, in a very basic way, this is the nutritional relationship in mycorrhizas. You have a plant that fixes carbon dioxide to produce sugars. Those sugars end up in the fungus, so that's a benefit for the fungus. But on the other hand, the fungus helps the plant acquire nitrogen and phosphorus from the soil in the different mechanisms. Now, this carbon is sugars, but it's also other things like amino acids, as I mentioned earlier. So this is your basic nutritional relationship in mycorrhizas. And again, this is exemplified in a better, I suppose, schematic where you have, this will be a pine tree here with the Ectomycorrhizal associations. This is very similar, if you remember, to the micrograph I showed you at the very beginning from my PhD work. So these hyphae going out into the soil or vice versa, basically it's a connection between the root tips and the soil. These are the conduits for nutrients that have been mobilized by the fungi. So particularly phosphorus and nitrogen, okay? Whichever way those nutrients are acquired by the fungus, they are conveyed to the plant, and the plant gives carbohydrates to the fungus, okay? Now, there are some very interesting, though, alternative associations, and let me see if I can, I wanted to close this thing. Anyway, sorry about that. I don't know how to do this. There are variations on the theme that are very interesting. So here you have, again, your main plant, let's say, that acquires carbon through photosynthesis, and it gives it to a fungus, and this fungus gives nitrogen and phosphorus back. However, there are situations where this same fungus, this same fungal body is actually connected, is also connected to another plant. And in some cases, this plant is not able to conduct photosynthesis. We call these achlorophyllous plants. So these are plants that don't have chlorophyll. For example, some orchids behave like that, and some other plants as well that are not orchids, like pine drops, for example. So here's a situation, a very interesting situation, because the fungus does not really, obviously does not produce sugar itself. So it actually conveys the sugar that comes from this plant to this plant, which is a parasite of the fungus, right? It's only gaining from the fungus. It's not giving anything back to the fungus. It's gaining both nutrients, nitrogen and phosphorus, and carbons, because it cannot fix its own carbon, right? It's an achlorophyllous plant. Some of the sugar can also come from the fungus decaying the litter in the forest soil, for example. So the fungus behaves both as a mutualist biotrophic fungus with this plant, but also as a saprophyte with the litter. Fungi can be very, very flexible in their ecological functions. Doesn't matter. This plant still remains a parasite of this fungus, right? So the interesting thing is that in ecological terms, we call this plant an epiparasite of this plant, right? Indirectly, this plant is parasitizing this plant. It's called an epiparasite. Epi means on top, from the Greek, I believe. So it means it's on top of the food chain, right? Because here's a primary producer, as it's called in ecology, is an intermediate, and then this plant is actually the final, the organism that benefits ultimately from this plant. So it's an indirect parasite of this plant. Now you may think this can occur between different plant species, and that is true. That's particularly obvious with some orchid associations, but it can actually happen with plants within the same species. So here's the cover of what is considered to be the Bible of mycorrhizal knowledge, this book, and it shows a seedling of Scots pine in this case, which is a European species, in a lab system where you can really see very well the mycorrhizal associations with the root tips of this pine seedling, and then the extramatrical hyphae that I mentioned earlier. So these are the ones that really expand the volume of soil that this seedling is able to explore, compared to the situation where it would not have this association, right? It's obvious right here. But the other thing that's obvious is that these other younger seedlings are actually tapping into this pre-existing system. And what this means is that if you think in terms of a forest environment, a mother tree dropping its seed, usually they're fairly heavy with trees. They drop very close to the mother, right? The seedlings germinate and they immediately find themselves in a highly shaded environment. So they're not able to produce a lot of their own carbon because they're shaded under the mother tree. Well, nature has found a way of providing carbon from the mother plant to its daughters and sons, so to speak, by way of this mycorrhizal network. So some of the phenomena that were described in the video that I showed at the beginning have to do with the ability of trees to behave as mother trees and essentially nurse their own offspring, which I think is a pretty cool concept. So I'm going to stop there. I think, I hope there will be questions. I'd like to put a plug in here for my own program. If you feel inclined to donate tax exempt to Ohio State and my program, you can go to my webpage, which is Department of Plant Pathology or .osu.edu/Bonello. And there you can find a way of donating to my program. And we always need money to do research. And so I'm not ashamed to beg at this point. So with that, I will stop sharing my screen and we can open it up to discussion.
Okay. Thank you, Enrico.
Thank you.
Great stuff. I mean, brings back some things that I probably had stored in the brain somewhere, but shake off some of the dust. So let's see. Yeah, we've got time and we have a few questions here. So Karen Lewis asks, what is known about how long it takes for the network to develop? I'm thinking about young woods and backyards.
So the networks are actually, for the most part, they are pre-existing unless the soil is deeply disturbed. That's one of the reasons that no-till agriculture is becoming more and more important for soil conservation, but also to maintain these mycorrhizal networks in the soil. So if you have a good soil, and I don't know, maybe who's the person?
Her name was Karen Lewis.
Can she actually converse with me?
Let's see. Can you find her on there, Marnie? And we can.
Yeah, I'm looking. I might be able to allow her to talk if she's comfortable doing so. I'm scrolling through all of our.
Or you can unmute everybody. No, really, I mean, it will be all right as long as they are patient and take turns.
Okay, Karen is allowed to talk.
Great.
Yeah,
Thank
So
Yeah,
there she is.
Yes. I'm thinking about, in a lot of developments, they scrape away the topsoil and put in a house and then try to plant trees. And if someone's trying to develop their yard, or I guess I'm just wondering how, I had two questions. Another question I had maybe is related is, can plants last a long time? I guess from those numbers, it looks like there's a percentage of trees and plants that don't function without this fungi. Is that right?
yeah, it's very, very small number actually. That for some reason people have not been able to document any mycorrhizal associations, but
Yeah.
those are not your typical. It sounds like you're talking about ornamental settings, like urban settings. Those are not your typical plants. We're talking about Chenopodiaceae, some other families.
Right.
Yeah. So yes. So new developments. That's a good question. For the most part, if you rebuild your soil with good sort of composted organic matter, that material will have inoculum in it that you will incorporate into your soil, and you don't really need to do any other kind of intervention to get mycorrhizae going. Okay. I
So would it-
strongly say again.
I was just wondering how quickly you might find this as a full network between multiple plants in a yard or a young forest or a place that's going back to nature,
Yeah, it depends. It depends on how disturbed the system is. If it's disturbed continuously, in other words, if your soil is mixed continuously, then you're continuously breaking those connections, right? Because these hyphae are very, very thin, very delicate, and so on.
Right.
And again, so going back to agriculture, that's why you have no-till, right? It's to maintain the structural integrity of the soil and the mycorrhizal networks. In forest environments, it's not an issue. Mycorrhizae are always there. In ornamental settings, again, it depends on what you do to your soil. If you keep mixing it, if you keep tilling it, and that sort of thing, then you continuously break those connections. But if it's only surface treatments, or if you add composted material to your soil every year, those connections will be well established and essentially will last forever.
Okay, thank you.
Sure.
Okay, so Adam Komar says, after reviewing peer-reviewed journal articles regarding possible parasitism of mycorrhizae on row crops, when applied in too great of concentrations as a supplement to fertilization at time of planting, are there examples of similar suspected cases of naturally occurring mycorrhiza on the landscape negatively impacting larger vascular plants such as trees?
That's a great question. You may remember one of my slides, I showed that in symbiotic associations, there's a gradient of associations between purely parasitic to mutualistic. And that is true also of individual species of mycorrhizal fungi, particularly in the Ectomycorrhizal fungi. So some may be more beneficial than others. And under certain circumstances, some can actually turn into pathogens, okay? It's also possible for some pathogens to behave as mycorrhizal fungi. There's been some, not a lot, but a few records of armillaria, for example, which is the shoestring fungus that causes root rot, most commonly in oaks, to behave as an ectomycorrhizal fungus. And conversely, it's been shown that the matsutake mushroom, which is a very prized species of mushroom in Japan, which is ectomycorrhizal, under certain circumstances can behave more as a pathogen than a mutualist. So there is some flexibility there, but by and large, mycorrhizal fungi are never pathogens. If you have application of mycorrhizal inoculum in conjunction with fertilization, you are nullifying the benefit of the mycorrhizal inoculation, because fertilization typically suppresses the ability of the fungi to infect the plants. It's almost as if the plant says, I don't need you, I have extra fertilizer here. Okay, so it's, as far as I know, I've never heard that under additional fertilization, these fungi can become pathogenic. This is news to me. I don't know if I understand your question correctly, but I've never heard of it.
Okay. Well, he has a follow-up that asks, is it suspected that the evolution of Mycorrhizae first functioned as a parasite rather than a symbiote?
It's possible. It's possible. I think that's all I can say. Now, bear in mind again that the nature of these associations is quite flexible. As I've just said, under certain circumstances, some fungi can behave more as mutualists than parasites and vice versa. It is an infection of a plant, after all. In fact, those arbuscules, which in general terms are defined as haustoria, right? These structures that grow inside the cells, those are also produced by some pathogens. So it's really the identity of the fungus that determines where that interaction is going. If it is a pathogenic fungus, it will hurt the plant, right? But it will hurt the plants through the same kinds of structures, these haustoria. So again, there's a lot of flexibility overall in these associations. So the question about the fossil record, I think it's quite difficult to answer. The only thing that we can say is that because the micrographs that I showed you there, for example, they also show these vesicles. It seems to suggest that these are arbuscular mycorrhizal associations rather than pathogenic.
Next one, CT Holcomb asks, is there a preferred way to introduce appropriate mycorrhiza into growing media for bonsai?
Ooh. I'm not sure I would know how to answer that question for bonsais. So these are, potted plants. In general, if you want to try and introduce mycorrhizal associations, you would do that by mixing them in the soil, right, beforehand. I'd like to give a word of caution, though, about the benefit or lack thereof of artificial inoculations in mycorrhizal associations, because it's very difficult to not have natural inoculum in your soil, even if it's very disturbed. We did some experiments 10 years ago or so, where we had a site in Worcester at the ORDC campus. There was essentially a trench that was dug for a pipe project. And we had soil that had been brought up from something like 6 feet or more below grade. And we tested that soil and we found Ectomycorrhizal inoculum in it. Okay. So that means that you can have mycorrhizal inoculum in general, even in disturbed soils. So I'm very skeptical about mycorrhizal inoculum for two reasons. One is that, as I said, very often you have mycorrhizal inoculum in almost in any kind of soil, even the most disturbed ones. The other one is that we discovered that we bought stuff off the shelf, and we discovered that most of it was non-viable, actually. So it was dead. That's one thing. The other thing is that if you look carefully, every time you buy mycorrhizal preparations, they also have fertilizers in them. they never come without fertilizers. And there is a reason for it, I think. And that is that the producers want to show that you have a plant response with mycorrhizal inoculation. But that response is actually due to the fertilizers they also have in there. So if you ask me if I wanted to buy mycorrhizal inoculant for my plants, I wouldn't. They don't hurt the plants, but I don't think they're worth the money.
Okay. Next one, Laura Capel asks if farmland is plowed yearly, does this disrupt mycorrhizal activity?
Yes, it does.
Okay. And then she had another one that can mycorrhizal networks exist in compacted earth, which you just kind of answered that to some extent.
Well, they can exist in compacted. Well, I guess it depends on the level of compaction, but they can exist in compacted earth, but they definitely wouldn't function as well as in a well aerated soil with good amount of organic matter.
Okay.
Where you have a good structure in the soil, not texture, but structure, where you have good soil conglomerates.
Okay. Roy Zinn asks, about when were these associations first discovered?
So these associations were first discovered, as far as ectomycorrhizal associations are concerned, in the 1800s, sort of a second half of the 19th century.
Okay. And Anonymous asks, what's the relationship between soil nematodes and mycorrhiza, benefit or detrimental to vascular plants?
So some nematodes actually feed on fungi. I suppose theoretically they could affect the mycorrhizal network, but I think the overall effect on balance is probably absolutely negligible, because the soil is full of nematodes, and if they were a real impact on these networks, I think we wouldn't have networks pretty much, because they produce obviously a lot of hyphal material in the soil.
Okay. Laura Faye asks, do you recommend the application of mycorrhiza when planting trees of any size, seedlings up to bald and burlap, which kind of talked about product, but any recommendations at all?
No, I mean, I personally wouldn't, as I said. You can, it doesn't hurt the trees.
Right.
You can get a response to the fertilizer in the preparations. The best thing you can do when you plant trees, and I think Kathy and Marnie will agree with that, is you prepare the soil. And if you can incorporate, if your soil does not have a lot of organic matter, you can incorporate composted plant material. That would be the best thing you can do. The saying that if you build it, they will come works very well for mycorrhizal associations.
Theodora Gregg asks, what about agricultural plants? Any mycorrhizal associations there?
Yes, all agricultural plants have mycorrhizal associations, including the legumes. The legumes, they have associations with bacteria, the rhizobacteria to fix nitrogen. They also have mycorrhizal associations. Remember, those are endomycorrhizal associations, not the ecto.
Tom asks, could Enrico make a couple of comments about glomalin, just a few basic descriptive intro comments or whatever detail he likes? Thank you. And the spelling on that is Glomalin. Just to make sure.
Glomalin is a protein that's produced specifically by the fungi in the Glomeres, and these are fungi that produce endomycorrhizal associations. And it's thought to be one of the most abundant proteins on Earth, I think. I'm not sure what your questions sort of implied. Generally speaking, you can come on if you want to have a little chat, specifically where you were trying to get to. But that's all I can tell you. It's used mostly to estimate the amounts of endomycorrhizal fungi in the soil. But if you want to expand more on your question, I don't know if
That was
Mark-
Ken or Kent.
I have to, no, I gotta go back. Let me, because I
Okay, cleared the question.
Tom Brochu, BROCH.
Okay. Got it. Okay. Tom, if you want to expand, if not, no pressure.
Can you hear me?
Yes.
Yes.
Okay, Glomerin is something I've just been fascinated by. I took soil science in the early 1970s, and obviously that was not something that was even known about in my day. So I think it was discovered in the early or mid 1990s. And I'm also a student of forest carbon. So in my retirement years, so Glomerin is something that has captured my interest. And I was just wondering if you could, I know it's related to the topic that we're really mycorrhizae. So anything you could add would be fine. No specifics, just general knowledge as I try to keep up with developments that occurred long after I was in school.
Yeah, I am not up to speed, as I said, the last few years. As I said, it's considered one of the most abundant proteins in soil. I think it also helps in creating soil structure. So in gluing together the soil particles into, what do you call them? Kathy, help me out, Marnie, maybe.
Not certain where you're
Thank you.
going.
That's fine.
Huh?
You can move on. Yeah, that's fine. I'm good.
Yeah.
Okay. Aggregates, as Kent said.
Aggregate, thank you.
Ah,
I need more
you,
caffeine
Kent.
this morning.
Soil aggregates, that's right.
And on that note, Kent asks, is it worthwhile to collect spores of mycorrhizal fungi and add them to compost used in soil remediation in urban tree plantings?
I don't think so. That would be a major endeavor. You wouldn't have to collect the spores. You could just mix mushrooms, I guess, into your composting pile. I don't even know what composting would do to those spores. It might inactivate them for all I know.
Oh, yeah.
I'm just thinking that you don't need to do that. Spores. So ectomycorrhizal fungal spores are airborne. So they're dispersed in air currents, and they're essentially everywhere. They just get deposited on the soil surface, and then they get into the soil by water percolation. I wouldn't do that.
That's a good answer.
Yeah.
Nicholas asks, are there specific connections with the mycorrhiza and tree seedlings and epiparasites? And are the fungi genera equally present either as echo or endo mycorrhiza?
I'm not sure I understand that question. Can you read it again?
So he says, are there specific connections with the mycorrhiza and tree seedlings and epiparasites? And are the fungi genera equally present either as echo or endo mycorrhiza?
Yeah, I don't really understand the question. So the epiparasites, the seedlings themselves can be considered epiparasites of their mother plants. Whether you're asking about, for example, the connection between a mother plant, a seedling, and an orchid, like a tree, its seedlings, and an orchid, that's certainly possible, all in the same network. And then I think the question was about equal proportions of ecto and endos.
Yeah, are the fungi genera equally present either as echo or endo mycorrhiza?
No, so well, it depends on the environment. If you are in a grassland, you have essentially no ectomycorrhizal associations. If you're in a mature forest, you have actually no endomycorrhizal association. So it really depends on the plant species that you have and the particular habitats that you find yourself in.
Okay. Nikki asks, how beneficial is adding purchased expensive powdered mycorrhizae to the plant roots prior to planting, which you've kind of hit on it. Not much.
It's one thing we don't have to worry about, right?
Right.
I
Forget
would
about that part.
It's a good thing.
Spend your money on better plant material than mycorrhizae.
better
Preventing
soil.
the soil, right?
Get better soil. Avoid creating the tub effect, in clay soils. Remove the burlap.
Cage.
The cage. Score your roots, because that helps with the root regeneration.
Yeah.
Note your planting depth.
Yeah, and
All that
even
good
with
stuff.
say with trees that have been in the ground for a while, if you have a very compacted organic matter poor soil, depending on the size of the plant, you can do air spading and actually replacing the soil with good stuff.
Okay. So Brian asks, how long after digging a hole to plant a tree or shrub does it take to rebuild those connections?
So I think, I don't know if anybody's ever looked at that, but I think that once the fine roots start growing in the spring, they get immediately colonized by those fungi.
Cool. I didn't realize it was quite that quick, but that's great.
Yeah.
Well, yeah, because the fungi are there, right?
Mhm.
And they can sense the roots. They sense the roots by way of chemicals that are released by the roots into the soil, amino acids and sugars in particular. They sense the chemicals and they grow towards them. It's called chemotropism, and so they sense it and they grow towards it and infect it.
Great.
Yeah.
Julie asks, does the same structure in root tips and fungi transfer sugar in and out, in/out, and nutrients in/out, or are there different structures for the transfer of different materials?
Good question. No, it's the same structures. You have a bidirectional movement of chemicals, essentially.
Okay. Perry asks, more than half of the forest land in Ohio was farmland 80 to 150 years ago. Are the mycorrhizae on those sites different from the mycorrhizae in forest sites that don't have that history of farming?
If you give me enough funding, we can try to find out.
On that note, there was a request in the chat for you to paste your website for donation into the chat if you want to do that. So folks have easy access to it.
Oh, that's wonderful. Yeah.
Okay. Let's see. Brian asked, do mycorrhizae also colonize/function in aquatic environments or just on land?
Sorry, I'm a little distracted here by the.
He's trying to put his.
No problem.
Yeah, well, I might as well do it now. Hold on a second. So, chat. There. As you can see, it's a give to Ohio State's. Oops, hold on. There you go.
There it is.
Can you repeat the question?
Okay, so do mycorrhizae also colonize/function in aquatic environments or just on land?
I think they also work in aquatic environments, but I'm not very well versed in that.
Okay.
Good question.
Yeah. Karen asks, beyond the exchange of sugars, nitrogen, etc., do we understand how the fungi transfer info about insects attacks?
Oh, yeah. Well, that's a very good question. I don't think there's a very good understanding of those particular signals. So we're talking about stress signals, essentially, that a plant can theoretically at least transmit through the mycorrhizal network. We know a lot more about the signals that go from plant to plant over land, so through the air, actually. Chemical signals that are produced by plants that are in distress get sensed by other plants, and then they activate the defense responses of those receiving plants.
Wow.
Yeah,
That's
I mean,
cool.
yeah,
Cool.
yeah.
Yeah.
So yeah, I think that the knowledge, I mean, they make it sound cool in that video, but I think the knowledge is very, very minimal, and I'm actually not aware of good information on that.
Another request to post the donation link again at the end of the presentation, so we'll do that. Anonymous asks, other than human impacts, what other external factors can be destructive to mycorrhizal networks, and thoughts on climate impacts?
Okay. So the networks are pretty resilient unless you have major soil disruption. So how can you have major soil disruption? If you have, for example, very hot, intense fires, you can affect the top layers of the soil to the point where you could affect the mycorrhizal networks, because you may or may not know this, but 90% of the root systems of plants and trees grow within the top foot of soil. And so in addition to damaging the plants, you can also damage the fungal networks. Other than that, I would say major soil movement activities. Yeah, I can't think of anything major in terms of disruption of the networks themselves. Now, in terms of effects of climate change, that's a lot more difficult to predict, I think. In part because the soil environment is much more buffered in terms of temperature than the atmospheric environment. There are there have been some studies looking at the frequency of mushroom production. Mushrooms are related to ectomycorrhizal associations in forest environments, and how that may be affected by climate change. Most of it, I think, has to do with species migration. In other words, as certain environment gets hotter, those species have a hard time growing in that particular environment and sort of migrate north. It's the same with other species of organisms like trees and so on, or the phenology of insect, development and the like. I don't know if there's very good information about our predictive ability to say what's going to happen to specific mycorrhizal associations with climate change.
Okay. Perry asked.
Sorry, maybe I can add that the effect of climate change may affect the mycorrhizal prevalence of certain mycorrhizal fungi by way of the prevalence of their hosts. That can definitely have an effect.
Well, and I was thinking when you were talking about the fire scenario, if fires increase in forests because of climate, I mean, there's another kind of secondary impact. So
yeah, if they become more frequent and hotter, then
right.
Of course you're going to have some effects.
Right. Okay. So Perry asks, are morels mycorrhizae?
Yes, they are.
I've been eating mycorrhizae all these years.
Mycorrhizal. Now, the mushrooms that you find in the stores here in this country are not, for the most part. So your regular, white mushroom, the portobellas, the shiitake, the oyster mushrooms, they're all saprophytes. They either grow, they're grown agriculturally in soil-like environments. That would be your typical agaricus, the white mushroom, or the portobello mushroom, or they must be grown on logs.
Mm-hmm.
That's true for shiitake and oyster mushrooms.
Right.
But in Europe, for example, where I'm from, the toadstools are consumed quite widely, and most of those are mycorrhizal. So you have species of Russula, Lacaria, Boletus, which is the porcini, and so on. Those are all my Ectomycorrhizal fungi, the toadstools.
That's interesting.
Some of them are toxic, of course, like
Right.
the Amanitas. You don't want to eat those. Those are also Ectomycorrhizal. Yeah.
Just remember that there are no old, bold mushroom hunters. Keep that in mind. So Laura asks, in raised bed gardens, vegetables that have compost added and mixed yearly, do mycorrhizae exist or help annual plants?
Oh, they're there, I'm sure. I would have no doubts that they develop yearly with the addition of that material, because you're bringing in the inoculum with that material.
Okay. Alyssa asks, as a naturalist, I am frequently asked about squaw root. Is this one of the plants that benefits from mycorrhizae?
What's that again?
As a naturalist, she says, I'm frequently asked about squaw root. Is this one of the plants that benefits from mycorrhizae?
Well, I don't know what squaw root is.
I'd have to go digging for even a good description. I don't know if I've got anything to help you with
Also
that.
called cancer root, bear cone, Conophyllus americana.
Yeah, I wouldn't know that, so I'm sorry, I cannot answer that question.
Yeah.
Sorry.
I'll look at a couple things, maybe we come back, but we're getting close to time. Nikki asks, would it be an exception to add mycorrhizae to potted orchid? Oh, did we already do that? No, to potted orchids. Is it already present in purchased orchid potting mix? If yes, I should add it.
That's a very good question. I don't know the answer to that. So most orchids either necessitate or greatly benefit from mycorrhizal association during seed germination and seedling development. And in fact, they are parasitic on the fungus, as I said, at least at the beginning of their life stage, if they are chlorophyllous. So if they produce chlorophyll later, they're still parasitic early on in their development. I don't know anything really about orchid production commercially. So I'm very sorry, I cannot answer that question. Interestingly, there is a student who's interested in working with me who would like to explore the use of fungi in orchid production. And he probably would have the answer to that question, but he's not with me yet. So
Okay.
Sorry.
So Laura Faye asks, can you clarify what the tub effect is? When we're talking about well, basically you dig a hole that is in enough clay that you create a tub that you just set the plant down in, the tree down in, fills with water, sits there, makes the tree unhappy. I always liken it to you dug a hole and you stuck like a wash tub, a metal wash tub down in the hole. And if you don't do something else with the soil, you've just set the tree into a metal wash tub in some respects. I mean, some water is going to move, but not much.
It's something that happens in clay soils.
Yeah.
High clay content.
Yeah. Or, you know, sometimes how you dig the hole, you may not have as high clay, but I have seen some that use like augers and stuff, and you end up with this smooth side that just becomes a barrier to going, the roots going anywhere. They're going to hit that and just not go through it. So that's the other way I've seen that kind of tub effect, is if someone's using an auger that they let spin in that clay content for a while.
Yeah, because the roots like to grow in between soil aggregates. And clay soils have very, very small air spaces, right? they tend to be compacted, water logged, and so on. And if you create this smooth surface into the hole, it's very difficult for the fine roots to go through that barrier. And also you encourage the accumulation of water and water logging in those tubs.
And most of the time you're not planting a tree that can tolerate standing water for long periods. So they're not happy campers.
All trees will not like standing water for a long time because it creates what is called an anoxic environment. So very low oxygen levels, which are very bad for the roots and therefore the whole plant.
Right. So Nicholas asks, can you address the interest of horticultural soilless mix producers in incorporating mycorrhizae into commercial greenhouse media?
That's beyond my expertise. I'm sorry.
That's okay.
Yeah, I don't work with greenhouse crops, so I wouldn't know.
So I've got someone who wants to know if they can contact you afterwards to kind of clarify some answers. Can we give your email?
You can email me, yeah.
Okay, we'll post your email address as well. Let's see, next one. Any opinion on whether mycorrhizae filter out microplastics?
No.
No. I figured that was what your answer was going to be. This kind of goes back to one of the other questions you had. Carol says, when was the first mycorrhizae found in the fossil record?
Back in that slide.
Yeah. Enrico.
No, those micrographs were for samples that were from deposits that were 400 million years old. So 400 million years old, very early in the development of land plants.
Okay.
So even that land plant, that land plant that's in the micrograph is a primitive plant, really. They were rhizomes. You might know or not know from your horticultural background that rhizomes are underground stems. And they were very primitive, excuse me, primitive in their anatomy. But it's still very interesting that you find these associations even at that early stage in the evolution of plants.
Okay, so last one that I have here is, what is air spading? Is that what you say helps making the tub? So the air spading doesn't make the tub.
Air spading is used on standing plants, so not to make the hole to plant a tree, for example. So if you have a tree growing in the landscape, like behind me here, like this one, or also growing at the same time behind Marnie, because we have the same trees behind us. We're in the same forest.
We're actually right next to each other now.
For those who don't know, this is a picture of, I think it's the Oval on campus. Yes.
Yep. There's the library.
There's the library.
Yeah, the main library down there. So if you have a tree like this and you want to improve the soil condition of that tree, you can use an air spade, which is a device. Essentially, it's a blower that produces high volume, high speed air jets. And the benefit of that is that you can blow away the topsoil without damaging even the fine roots of the tree. And so if you want to replace that, the soil that the topsoil that the tree is growing in, you can use an air spade and then backfill with good stuff. So that's done horticulturally these days. It's one of the best ways to improve the soil condition for standing trees, much more than some arborists will suggest deep fertilization or deep watering, or what do they call it? The mulching, where they drill holes.
Vertical mulching.
Vertical mulching, right? Yep. So they drill holes and then they put mulch in the holes. Air spading does a much better job, much more extensive improvement of the soil condition for the whole tree.
There's a tree that is north of Lane Avenue, south of the 4-H Center, that is showing impacts of all that construction in there. And we just had that discussion on our tree campus committee this week about bringing somebody in with an air spade, because we figure it's the compaction from that underground stuff they were putting in there on the corner that has impacted a section of that tree. And so they're going to bring an air spade in to try to minimize or, you know, try to deal with that compaction issue. So
let me just show you very quickly what an air spade looks like. So it's essentially it's a blower.
mhm.
It's just that it has a very long tube that's connected to a compressor, right? And then you have this long tube with a nozzle that produces high velocity airflows to blow off all the soil from around the tree.
Yeah, they're fascinating to watch. I
Yeah.
remember the first time I saw one was out the tree they did out in front of Howlett. That was the first time I had
I mean,
seen
they make a
one.
mess, but
Yeah, they do.
Yeah, so does the soil on top just like poof, like,
yep,
Yeah,
blows
Wow.
away.
yeah,
huh?
yep. And they just keep going down and down, and you just keep revealing roots at all the levels. I mean, it is fascinating to watch.
Yeah, you can see the whole root structure, the whole root
Right.
system structure. For
Yeah.
most of it.
Yeah, when they did that, I don't, was that the hackberry in front of Howlett that they did?
There you go. There's another good definition there in the chat. Air spade is
Mhm.
like a power washer that uses air
Uses
instead
air
of water.
instead of water.
Yep.
Good
Thanks,
deal,
Jamie.
Jamie.
Good.
Okay, I think we need to let you go.
Yeah.
Yeah.
Sorry, folks, that we don't get him for another half hour, but I did post his email in the chat, and we will, so you can access that. And then I need to get you the link for the credits. So
Do you
thank you,
guys-
Enrico.
You're welcome. If this is sort of evaluated or rated by the viewers, I'd like to hear back.
Okay. Let me put in Marnie, stop recording, and we'll